go live.
Okay.
All right, there we go. All right, so I think we're live now. Yes, so here we are. I'm here with Christine Massey. We are going to be
Okay.
talking about, all right, oh, if you just, on your end, if you just turn down the volume on the stream, it's coming through on my end a little bit. All right,
That's right.
so I think we're live
I
now.
can put a headset on. Will that
Yes,
help?
so here we are.
I can
I'm
try
here
the
with
headset.
Christine Massey.
I don't know
Okay,
if that helps.
we are going to be
Okay.
talking about,
Improvements.
all right, oh, if you just, on your end, if you just turn down the volume on the
Sorry, should
stream,
I have
it's
this
coming through on my end a
ready?
little. All right,
That's right.
so I think we're live
I
now.
can put a headset on. Will that okay?
Yes,
Help?
so
Let's try
here we are.
this. I can
I'm
try the
here with
headset.
Christine Massey. Okay,
Is
we
that
are
better?
going to be
Okay.
talking
Oh,
about,
sorry. I don't have it working yet.
oh, if
Oh,
you
sorry.
just,
Shoot. Yeah,
on your end, if you just turn down the volume on the
sorry. I should
stream,
have
it's
this
coming through on my end a
ready.
little. All
Tell
right,
us what it's
so
a
I think we're live
headset. I
now.
can put a headset on. Will that okay?
Yes,
Help?
so
Let's try
here we are.
this. I can
I'm
try
here
the
with
headset.
Christine. Oh, there
Do
we
you mind
go.
saying something, Dan?
Okay, that works.
I'm not hearing any. I'm not hearing you. Let me just try it one more
That's
time.
better.
So I'm
That's
just
better.
gonna re-plug it.
Can you hear me?
Phone. Let me see. Hello?
Can you hear me?
It usually works.
It's okay.
Why
You
is
can
it
take it
not?
off. Never mind.
Sorry about that. It usually works.
I think I fixed it. It's okay. All right. Anyway, so do you have the stream up on your computer?
I see you. Is that what you mean?
Do you have? So I'm gonna be putting the papers up on a screen that'll be on the live
Okay.
stream.
Okay, great.
So anyway,
Does it sound okay now?
yeah, yeah. I think the sound's fine. So anyway, I'm here with Christine Massey, and so we are here because there are people who think that SARS-CoV-2 and viruses are not real and don't cause disease. Christine, you are one of them. You believe that SARS-CoV-2 does not exist and does not cause disease, correct?
Yeah, I would say it's never been shown to exist, and of course, if it hasn't been shown to exist, then it can't be shown to cause a disease. So that's my position. Yeah.
Okay. So, we agreed to have this discussion, and in this discussion, we said that we would discuss two papers. And those two papers, one of them is this one that I have on the screen, a novel coronavirus from patients with pneumonia in China, 2019. And the other one is this one, in situ structural analysis of SARS-CoV-2 spike reveals flexibility mediated by three hinges. So those are the two papers we are going to be discussing tonight. The evidence within them definitely demonstrates that SARS-CoV-2 is a real virus that exists and is associated with disease. So this is going to be open form, right? We agreed on the ground rules beforehand. So let's just get started, shall we?
Can we just clarify, like, just so your audience knows, we agreed we're sticking to the evidence that's actually in these two papers.
Right. It's only the
Right.
evidence that is in these two papers.
Right.
So, you want to get started?
You want me to go first?
Oh, well, I was just going to start going through this paper.
Sure.
Okay. So, this is the first paper that isolated SARS-CoV-2, the virus, and characterized it. So, I'll just start going through it, and you can let me know if I say something that you want to object to or what have you.
I know a way, I mean, I have a suggestion
Okay.
for the way I look at it, one way to simplify it would be to start with sequencing, even though it's sort of jumping in at the middle,
You want to go to
because
sequencing. Okay.
my position is neither of these papers found an intact genome. And we were trying to determine, like, we're not looking at disease causation in these papers because they're not even testing for that. So we were just looking at whether the alleged SARS-CoV-2 particle is actually shown to exist and has been purified.
Mm-hmm.
That's what, right? So the way I'm looking at it is, if they didn't even find an intact genome, you can't say that they actually showed that the particle exists, because the alleged genome, is part of the alleged particle.
Great. Okay. So we can start
So
with the sequencing. So
sequencing.
we go down to viral genome sequencing. Okay. So
Yeah.
they
You're in
describe
the
how.
methods. You're in the methods section.
Yep. They describe how this
Okay.
is done.
Sure.
So they had patients who were presenting with pneumonia symptoms, and these pneumonia symptoms were unexplained. They tested for every other known pathogen that they could in these patients to try to determine what was causing their symptoms. They could not come up with an answer. So they ended up trying to see what else might be in there causing the illness. So they extracted RNA from bronchial alveolar lavage fluid. This is fluid from the lungs, and culture supernatants. Okay. So they cultured the virus in cells, and they also extracted RNA from culture supernatants. And then they used a combination of Illumina sequencing and nanopore sequencing to characterize the virus. They obtained 2 near complete genomes and 1 complete genome from these samples.
So I don't, I can let you talk, Dan, if you like, and sort of say what you have to say about sequencing, and then say what I have to say. I don't really want to interrupt you. That's
so
okay?
so that's what they did.
Okay.
Okay, and they found again 2 near complete sequences and 1 complete sequence, and these sequences mapped most closely to the sarbecovirus coronavirus family, most closely related to, at this time, SARS-1, about 86.9% nucleotide sequence identity. So, do you have objections to this?
I have, I'll just give my little spiel then. So we both agreed they took RNA from the valve, the bronchio, I don't know how you say that word, bronchioalveolar lavage fluid,
Mm-hmm.
and also from the cell culture supernatant. So that's where they mixed a clinical sample with a cell line. They don't mention fetal bovine serum or toxic drugs in this paper, I don't think. Normally those are used, but anyway, we
Well,
know
those were used for cell culture, but they're not toxic, so we can get to that later if you like.
they
But the
use the
sequencing.
fetal bovine serum to feed the cells.
Mm-hmm.
So
Well,
right, so they
sort
took
of.
RNA. So what I'm getting at is they're not taking RNA from purified particles. They're taking it from the clinical sample, which is the valve fluid, and they're also taking it from cell cultures, which has valve mixed in with the cell line. And so it's not
So
coming from any particular particle. They have a soup basically of RNA from, they would have RNA from the human, from the cell line, any bacteria or
mhm.
fungi that happen to be in there.
So
And
from the supernatant, that wouldn't be the case. They would only have RNA from particles that are small enough to be viruses.
well, how do you figure that? Because they're saying they took the RNA, I'm just reading from the viral genome sequencing section.
Mm-hmm.
RNA extracted from the BALF fluid and culture supernatants
Mm-hmm.
was used as a template,
Yes.
right?
So they took
So,
so they took RNA extracted from cell culture supernatants to clone and sequence the genome.
so
So,
that's what they
but
say,
but in
but
either case, right? In either case, this method of Illumina or nanopore sequencing after RNA extraction, it's not going to matter if there's other things in there. You can still sequence a complete genome from an impure mixture.
So this is where I disagree, Dan, because our purpose here was to show, not to assume that there's, this virus has already been shown to exist and that the genome is in a database somewhere.
Oh, it's not assuming
This was,
anything.
I'm sorry?
It's not assuming anything.
Okay. Well, so what they're saying is they extracted the RNA from the valve and from cell culture supernatant. Now they hadn't purified, it's not coming from purified particles, it's coming from the cell culture supernatant. And they say that they got sequences, reads, and they assembled them into contigs.
Mm-hmm.
Right? So they're not saying that they found an intact genome. They're saying sequence reads were assembled into contig maps. And in bracket, they explain that's a set of overlapping DNA segments. So they found many sequences. And I'm actually just going to run ahead to the very next page where they're into the results. You get a little more detail. So they say RNA extracted from the bowel fluid from the patients, yada, yada, more than 20,000 viral reads from individual specimens were obtained. So they're getting thousands of reads, and then they're assembling these into what they call contigs. And then from there would be more assembling again. So it's not, and they also mentioned too, then they did specific primers were designed, and rapid amplification of complementary DNA ends was used to fill genome gaps. So they're saying right there, they didn't find an intact genome.
Yeah,
They've got thousands of bits and pieces, and they've put them together in a computer.
So this is how next-gen sequencing works. So do you have an understanding of how the method actually works, like what happens in order to obtain those contigs and the genome as a whole?
I don't know all the nitty-gritty details. What I know is what they're explaining here is that they found thousands of reads and they put things together.
So
the computer
you
put things together for them.
Do you think it's important to know the details of how next-gen sequencing works?
No, I don't, because if you're putting things together, that's not the same thing as finding an intact genome. So to me, the details are neither here nor there. If you can't find an intact genome, you can't say that there is an intact genome. And what you've done is make something. And so at that point, also, I should have mentioned something else from the beginning. So I'm going a little off topic here. I apologize.
So it's okay. We can focus on the sequencing because it's important.
Okay.
So
sure.
I have on screen right now in the live stream a little video explaining Illumina sequencing. Okay.
Okay.
So I'm gonna play it. Can you see it over the live stream?
I don't see anything right now. I just see you.
Okay. If you go to the YouTube link, you'll be able to see the live stream, and you'll be able to see what I'm showing you. Because this is important. So if you want to, if you want to say that these methods are invalid.
Sorry, Dan, I'm having a, Dan?
Yeah.
Sorry to interrupt you. I'm having a little problem because I'm getting the audio from both sources now and it's not lined up.
It's okay. What you can do is you can mute the window on YouTube, or you can mute the little icon bottom left. You can mute that and just listen to the call.
Okay, I'm muting it. Okay, that's good.
It might be a little delayed on the stream, but so it's important to understand these methods if you're going to say that they're invalid and can't show a fully characterized genome. This is how next-gen sequencing works. This is how we sequence all genomes today. This is a fast method to do it. It works on the exact same principles that the ways people used to do it by hand decades ago. So this is just a faster way to do it by hand, so that it doesn't take months and months to sequence a single genome. So here's how it works, okay? So you have lots of different. This is showing the setup before what I have here. So it's showing lots of strands of genetic material, DNA, at this point, that have been attached to a glass slide. Okay, that's called the cluster generation part, where they create lots of different clusters of each individual strand in one particular place. So that in this part, when the actual reaction, the sequencing reaction, is happening, you have all the nucleotides that are tagged with fluorescent molecules. And when they bind in the sequencing reaction to their complementary base pair, they fluoresce. That fluorescent tag that is attached to each nucleotide is released, and you get a burst of fluorescence. So that's what it's showing here in this physical chemical reaction. Okay? So skip ahead to what this actually looks like on the flow cell. So this is the entire glass slide chip with all of your nucleic acid on it. And when this reaction is happening, it looks like this. You have lots of different spots of fluorescence coming up all at once, representing the different nucleotides binding to its complementary base pair. Okay? So this happens on a big scale. And then the last part I want to show you, people are still trying to join, but the last part I want to show you is this here, because once you have all of those reads, which are measured by the fluorescence, which is the chemical reaction, you have something that looks like this. Okay? You have lots of individual reads, and these reads are going to overlap with each other in ways that can only fit one way, right? These overlaps are not random. So each piece is not a complete genome, but that's how this sequencing works. You sequence lots of little parts, you sequence the genome lots of little parts at a time, and then you can put them all together by looking at where they overlap. Again, you can do this by hand. This is how people used to sequence by hand decades ago. Now it's just faster. So this is real physical sequencing. It's not just made up in a computer. This is how the reaction actually works.
So, okay, Dan, so I understand this is how people have been doing things. But again, I'm arguing that it's not an intact genome being found. It's things are being put together and there's assumptions being made because, just because you have two sequences and there's some, maybe you have ATC here and ATC here, it doesn't mean they actually were,
Oh, but it's not,
part of a
it's,
longer whole. That you don't know that, right?
well,
Maybe they
we
were this
do
way, maybe
because,
they were this way,
so
maybe they were nowhere
hang on,
near
hang
each
on,
other.
hang on, Christine. So it's not just ATC here and ATC here. It'll be strings of dozens or even hundreds of nucleotides overlapping
Yeah, I understand.
over several hundred reads of that one area, or thousands of reads of that one area. And so the odds of all of those reads overlapping with each other for dozens and dozens of nucleotides, what are the odds of that? I mean, it's astronomical that it would be random.
Well, again, this is based on probabilities, and I mean, it's basically a black box thing too. It's not like we're all sitting here actually looking at these things and deciding for ourselves. We have to take the author's word, but this is all based on
Oh, but you don't have to take the
calculations,
author's word.
probabilities. It's not the same thing as finding an intact genome.
Well,
Um,
it really is, though, because not only can you do this once, you can do it multiple times in multiple different labs all over the world, and that's what was done. And you can also do something called nanopore sequencing, which they do mention here. Do you
yes,
understand nanopore sequencing?
I don't have a great understanding. The way I heard Jay Cooey explain it was that you can pass a whole bunch of sequences through at the same time and it detects them all as I don't know, it detects them a whole bunch of things at once, basically.
Okay. I mean, again, do you think it's important to know how things like nanopore sequencing works?
Not particularly, no, because again, like I said, my position is if you don't either way, it's not being found intact, and working off of probabilities, because there's uh, a lot of sequences repeating, you still don't know for sure how things fit together.
Well, we do
Um,
because
I
we can measure that genome, we can see how it fits together, and we can predict what the proteins are going to be. And we can then see that the proteins actually match that genome.
Dan,
Hmm.
so in this paper, there is no protein analysis.
Oh, that's in this paper. In situ
Yeah, but that's
structural
a separate
analysis.
paper with that's a different substance. Those two papers don't go together.
Oh, but they do. I mean, that
No, they
it segue
don't.
it segues. Our conversation just segued into the data that
These
is
are
shown in this paper.
these are two separate studies. In the other study that we're going to discuss, they tell you where they received their so-called isolate, and it's
Hmm.
not from these authors. It's from a completely different paper. So
It doesn't matter.
they're
It's the same virus, isolated
no,
from a patient.
that's an assumption. We're here to show if we can establish that the particle that is called the virus even
Okay.
exists.
Okay. So if it was an assumption, right? Let's assume that it was that they just were sent something that is not SARS-CoV-2. Okay. What would
Wait,
happen?
sorry,
What would happen
can
in their
we,
data?
sorry, can I just, before we go on to the other paper, can we, can we just establish a couple things about this first paper? So you agree that there's no protein analysis in this first paper?
They didn't look at proteins. No, they
Right.
looked at cell culture. They looked at genome. They looked at electron microscopy, among some
So we,
clinical findings as well.
okay, so we agree on that, and we agree that they didn't actually find an intact genome. They did something based on
Oh
probabilities
no, we
and
disagree on that completely because next gen sequencing is how you sequence a complete genome. Do you? I mean, if someone, so if God forbid, if someone broke into your house and left their DNA in your house somewhere, cops came, swabbed the place, they found a DNA profile, and then your neighbor's house gets broken into. They Cops come and do the same thing. They swab, find a DNA profile, and they find that it's a match. The same person broke into your house and your neighbor's house. And then they catch that person and they match their DNA to those crime scenes. Take that person to court and they say this genome evidence proves that this person broke into both houses. Would you accept that in court? Would you accept that that person should be convicted?
well, if you're describing a scenario where intact something or other was found, possibly, I mean, there's all the issues around, finding bits, finding DNA doesn't prove that somebody killed somebody or what have you. But again, we're not
So you
talking
wouldn't
about finding
you
something
wouldn't
intact.
you would think that person might be innocent if their DNA was matched to both yours and your neighbor's crime scenes and they had never been in your house
You're
before to
you're
your
talking
knowledge.
about a scenario where intact information is found,
It's the same
possibly, but
techniques.
that's
It's the
not what
same
we're talking.
it's the
Well, if
same
that's
kind of
what's
techniques
used,
except this is better because you actually sequence the genome.
again, I disagree. But again, also, I want there's something else to share with you. So the there's an investigative journalist by the name of Torsten Engelbrecht, and he wrote to the author. Well, actually, I'll just read from page 728 in this paper under the section Transmission Electron Microscopy. It says supernatant from the human airway epithelial cell cultures that showed cytopathic effects was collected, inactivated with 2% pure formaldehyde for at least 2 hours, and ultracentrifuged to sediment what they're calling virus particles.
Hmm.
So I just want to point out a few things. So they're not finding something directly in a clinical sample, they're only working with cell cultures where they obtained cytopathic effects. And they're saying that they ultracentrifuged to sediment virus particles. They don't
Hmm.
say that they purified virus particles. Torsten Engelbrecht wrote to the author, the last author who's listed, I don't know how you pronounce the first name, but it's Wenji or something, Tan, Tan. And those emails, it's on his website, it's also on my website. People can find them. And he asked, did you purify? You know, you say that you sedimented virus particles. Did you actually purify them or did you only sediment them? And the answer came back very clear explicitly. We did not purify. We only sedimented.
Hmm. Yeah, but
So
so
the author has admitted that they did not purify. We don't have any protein analysis in this paper, and obviously you and I disagree on the, on the sequencing part.
sure. So can you really justify why you disagree about the sequencing, though? I mean, what about the method is flawed? Because it's not just based on probabilities, it's based on empirical findings that you analyze after physically sequencing the DNA that you find, the RNA that's turned into DNA.
We said we would discuss what is in these papers, and what is in this
And that's the
paper
method.
is
That's the
what it
method.
is. It's, you just showed something where a bunch of things are being, there's a bunch of reads and they're putting stuff together. I mean, it's not the same as finding something intact.
It
They
is,
didn't
though.
purify particles. They admit they didn't purify particles. What I would want to
They don't need
see
to
is
purify them.
particles. I'll just say what I would like to see:
Hmm.
particles found directly in a clinical sample, not in a cell culture, and purify them and do a visual check, electron microscopy, and show, there should be lots of these particles and
Hmm.
only one type of particle. And then extract the RNA from that particular particle and do Sanger sequencing, find the genome actually intact. That would be completely different, but
So,
that's
again,
not what I'm
Sanger
seeing
sequencing,
here.
Sanger sequencing also sequences in steps. You know that, right?
I understand. I've heard you have something and it chops things off a bit at a time. I don't know how they do that, but anyway.
You really might want to understand the methods, Christine, because it's kind of a problem if you're going to just object to what they're doing but not really understand what it is that they're doing. I mean, if I went to like a pilot and said that I don't trust them to fly this plane, and they said, "Okay, well, here, let me show you. I can show you like how the wings work." And I say, "Well, what's a wing?" They'll be like, "Oh, well, you can't tell me that I'm not qualified to fly this plane because you don't know anything about flying." I mean, you have to understand the methods, right? If you want to say that they're not valid. I think that's fair, right?
I disagree, Dan. I don't see any need to go into details when clearly they haven't found something intact. They haven't demonstrated that they found it intact, and they haven't demonstrated that the stuff that they used to make the genome came from any particular particle, because they didn't purify particles.
So again, they don't need to purify viral particles. So they obtained them in a relatively pure form by ultracentrifugation and cleaning up the supernatant and so on and so forth. Uh, those methods are going to get rid of cell debris, it's going to get rid of bacteria, it's going to get rid of fungi. They mention that in their methods, that all this was done to remove cell debris.
Um,
So,
Dan,
so
sorry to interrupt.
go ahead.
So um, there could be all sorts of RNA in that left over. There could be bits of RNA from the human, from bacteria and fungi. I
So
understand they would have removed larger particles, but there can still be RNA from who knows what sources.
So why did we find RNA that doesn't match to anything we knew before? Where
It
did it come
doesn't
from?
say that it doesn't say that they checked for that.
Yes, they did.
And
So
plus, looking in a database and you say, okay, you have thousands of reads, and then you say, okay, these ones we recognize from our database, so we take them out. That doesn't tell you that the stuff left over are all parts of some longer whole. Like maybe there was no longer whole. It's
it,
just an assumption that's being made that we have to put things together and create some longer thing.
again,
I'm sorry.
it's not an assumption, and I think you're saying that because you don't really understand how the method is done. I think there's a
Well, Dan,
fundamental lack of understanding there. But
how,
let me just read from the paper here. So so they state before they do this whole thing that the patients came in with their symptoms and they essentially could not make a diagnosis. They extracted nucleic acids and they tested for viruses and bacteria using several different methods in accordance with their manufacturers and tested for viruses, bacteria. They got negatives. They couldn't find any known pathogen that could be responsible for the patient's symptoms. They also,
that just,
when they took the human airway epithelial cells that they used to grow the virus, they screened that cell line because it's primary cells taken directly from a human. They screened that cell line for all known pathogens and found it to be not contaminated. And so when they added this cleaned up sample, this centrifuged sample that was taken directly from a patient, and the only things in there that are infectious could be viruses, they obtained a new viral genome that they had never seen before.
Dan, could I
It
interrupt you,
was not
please?
assumed. It was
Dan,
empirically measured.
could
Oh,
I
go
please
ahead.
interrupt you?
Go ahead.
Um, so I forget how many, oh, it says right here, samples were analyzed, this is on page 728. Samples were analyzed for 22 pathogens, 18 viruses, and 4 bacteria.
Hmm.
Um, it's not a reasonable assumption to say, okay, if we don't find any of these 18 viruses, alleged viruses that we're testing for, or
Hmm.
any of these 4 bacteria, then there has to be some other virus that made the person sick. There's
So
lots of things in this world that make people sick, and there's no reason to assume there was even a pathogen to find in the first place. And
so
certainly just testing for 18 alleged viruses and 4 alleged bacteria doesn't tell you that the only other possibility was that there was a new coronavirus causing the person to get sick.
so again, it was not assumed that it was a coronavirus. They thought that it must be something else because they were testing for everything that they knew could be causing this patient's symptoms. They knew it wasn't fungal, they knew it wasn't bacterial, it wasn't for any of the common pathogens that would normally be causing those symptoms in a patient. So they
Dan,
did the next sentence in an unbiased high throughput sequencing previously described. Again, it's not biased. It's not making an assumption. It's just it's asking what is there. And they found a new viral genome.
where you're declaring that there's a viral genome, but there's nothing in this paper. We already agreed to demonstrate that there's actually something, there's no testing of causation. There's no valid independent variable. There's no animal studies. There's not even a protein analysis.
Yeah,
And again,
that's not part of
there's
part of
many
this
things that,
paper, but
right. And we're
I told you
discussing,
I told you we could
we're,
discuss other papers and you didn't want to do that. But so this is what we're
no,
discussing and
well, then,
this shows a viral genome existing in a patient with an unexplained illness.
right. So we agreed to discuss 2 papers, and you said that both of these papers show the existence of the particles and that the particles were purified. We have the
It
author
was purified in this paper.
expressly admitting that they were not purified, and there's not even a protein analysis. I think we can stop here because, I mean, I'll just make one final point, and we could go on to the other paper. Um, Alleged pathogens are not the only thing that can cause disease. There's many things that can make people sick with respiratory illnesses. There's many factors that affect health. There's nutrition, there's exercise, sunlight, fresh air. Were you exposed? There's all kinds of toxins in our environments these days. You could be taking toxic medications, wearing masks. Have you had something shoved way up your nose, a swab? I mean, electromagnetic influences, stress. These are all things that cause disease. So it does not make any sense to assume that, oh, there must be a new virus in here, and all these bits and pieces of leftover RNA come from a virus genome.
Okay, so yeah, lots of things can make people sick. However, in this paper, it is showing unambiguously that a new virus exists in a patient with an unexplained illness. Several
Yeah, what
patients
is
with an unexplained
even if
illness.
even if we put aside the issue of not having purified particles, what is there in this paper to show that there's any particular particle causing the person to get sick?
The sequencing, the cell culture, and the electron microscopy.
Dan,
Those all
does
demonstrate that
sequencing
it is a virus that is in
does
this patient with an unexplained illness.
does sequencing prove causation of a specific particle?
We're
That
not
a
we're not
specific particle causes a disease?
Christine, we're not talking about causation of disease in this paper.
Right. So you're saying things that have not been actually established in this paper. It does not show that there's a new particle that caused any
It
illness.
shows a new particle associated with a new unexplained illness.
So I think we can stop here because we don't agree, and we're just going to keep disagreeing with each other. I mean,
So
we
in
did it.
summary, you don't think it's important to learn the methods that you're saying aren't valid.
Well, we didn't come here for you to educate me about sequencing, and we
I did.
already agreed that there's no protein analysis in this paper, and the
It's not
authors admitted
necessary.
that they did not purify particles. So
It's not
if they didn't
necessary.
purify any particles, they couldn't have purified SARS-CoV-2 particles.
It's
And
not
if they haven't
necessary.
even shown that there's a SARS-CoV-2 protein, obviously, like we have not shown that there's a SARS-CoV-2 particle, because you still need to find, even if I gave you the genome, which I'm not, but even if I did, that would still leave the whole question of the alleged spiky protein shell that is supposed to surround the alleged genome.
Yeah.
So I think we can stop here because we don't agree.
So
And every
let me just read another section of the paper real quick. So this is talking about the cell culture portion. To determine whether virus particles could be visualized in 2019 nCoV infected human airway epithelial cells, mock infected and virus infected human airway epithelial cells were examined with light microscopy daily and with transmission electron microscopy 6 days after inoculation. Cytopathic effects were observed 96 hours after inoculation on surface layers of human airway epithelial cells. The cytopathic effect was a lack of cilium beating, seen in light microscopy. They also infected Vero E6 and HUH7 cells, which the Vero E6 and HUH7 cells showed different cytopathic effects on a different timeline. However, all three cell lines were able to permit replication of this virus. They were able to see viral-like particles in transmission electron microscopy consistent with coronaviruses, and they were able to identify a new coronavirus genome. So you we disagree on the genome sequencing, but I think that we've shown that you aren't really interested in learning how that works or what it really shows. You don't accept cell culture as a valid method, correct?
Well, for one thing, Dan, when they do these cell culture
Mm-hmm.
experiments, we don't have, they want to attribute what happens to the cells, the cellular breakdown, which they call cytopathic effect. They're attributing that to an alleged new particle, but they didn't use this alleged, they didn't even find this particle, and they don't have a valid independent variable. They're not using purified particles. They're putting their the valve sample into the. I mean, it says right here in isolation of virus, the valve fluid samples were collected in sterile cups to which virus
Mm-hmm.
transport medium was added.
Yep.
Now they don't say what they used for virus transport medium.
It's just a
The
buffer.
CDC's recipe includes fetal bovine serum and toxic drugs, but we'll just give them a pass
So
on
so
that.
which so which
And
drugs?
sorry, can I just finish this, please?
Uh,
Samples were then centrifuged to remove cellular debris, and
mhm.
then that supernatant that was left over is inoculated onto the cell line. So they haven't purified particles. They don't have a valid independent variable, and to attribute causation to a specific particle, you have to have that particle as your independent variable. Otherwise, like, this just isn't, it's not a valid experiment, and it's not showing disease because it's not an animal experiment. It would just be, they put this centrifuged valve sample into the cell line, and then they wait several days, and if they see cytopathic effects, they blame it on this alleged virus, but they haven't used this alleged particle as the independent variable.
So
So I think we
what
disagree on the
other?
isolation part as
Yeah,
well.
yeah, I know we do, but so
Yeah.
you said toxic drugs. What toxic drugs are used in cell culture?
Well, typically they use what, amphotericin. They use antibiotics and antifungals, amphotericin, gentamicin.
Do you know what concentrations they use those at?
Not off the top of my head, no. But they don't. They never. They don't do controls. The only way to do a proper controlled experiment is to have everything the same in the experimental group and the control group. The only difference being the particle that you're interested in, but they
So
never
that's
have purified particles.
that's the
We
mock
haven't even
infected.
established there's a no protein analysis even in this
Okay,
paper.
okay.
So
So you keep saying the same things, but
yeah, nothing
the
there.
mock infected is the control here. But so
I think
when I culture cells on a weekly basis, so I use a media that contains fetal bovine serum and penicillin and streptomycin, among other things. But I use that media to just grow my cells all week. Do you think that those cells should be showing cytopathic effects because they are growing in the presence of antibiotics?
what I know is scientific
Can you
method
answer the
requires
question,
an
yes or
valid
no?
independent variable,
Christine,
Dan. You can't just, I don't make assumptions.
can
I
you
want to
answer?
see
I'm asking you a direct
valid
question here.
experiment.
Christine, I'm asking you a direct question.
What I know is
Would you expect those cells to display cytopathic effects grown in those conditions?
in conditions with,
Antibiotics.
I want to see a valid controlled experiment. Dr. Stefan Lanka did a controlled experiment
No,
and he
he didn't.
showed how you
But
can
we're
get
not talking about
cytopathic
Stefan
effects
Lanka. We're
without
talking
adding
about this
any,
paper. We're
without
talking
adding,
about this
yeah, I
paper.
know, but now
So,
you're going off into a hypothetical
no,
situation, and that's not what we're here to
Christine,
discuss.
Christine, I do this every week. I grow cells every week.
That's not,
It's very important to this paper because they are using cell culture methods that are identical to what I do every week. So,
Dan,
would you expect cells just growing all week, nothing else done to them in the presence of antibiotics? Would you expect them to have cytopathic effects? Can you please answer that
Dan,
question?
no, we're not here to discuss hypothetical situations. We're here to discuss what is in these
It's not
papers.
hypothetical. The answer is no. Cells grown in the presence of antibiotics do not show cytopathic effects if they're maintained properly. Because, and you can see that in this experiment with the mock control. Okay?
There is not a
That's
valid independent variable,
That's what
Dan.
is
I
on
mean,
the independent
I
variable is
think
the
we can,
sample. But if
I
you
think
see
we
on the
can
screen
just,
here, the mock control, the cells are fine, their cilia are beating, and in the infected cells, you
Dan,
see cytopathic effects.
what is the purpose of doing that experiment and observing cytopathic effects?
You're observing the virus replicating and its effects that it has on the host cells.
So you're attributing causation to a particle that hasn't even been identified and
It
is
has
not
been
an
identified because
independent
of the
variable.
sequencing and the electron microscopy.
Okay, I think we need to just stop here because we don't agree. Everybody can read these papers and see for themselves.
They can indeed.
So I think we might as well stop there.
So you don't agree with sequencing because reasons. All right, so the reason I wanted to include this next paper is because it completely is in 100% agreement with the sequence that scientists all around the world have measured at this point. So what it does is it uses cell culture methods which, like we're discussing, or like how I'm saying, are valid. There's no reason to discount cell culture methods to grow a virus which needs a host cell, and then they are purifying it using sucrose gradient, using sucrose ultracentrifugation, and then they are imaging that virus, what they get out of that centrifugation, in cryo-electron microscopy. Cryo-electron microscopy is really important here because it allows us to see these molecules on a near atomic resolution. It means you can see the individual atoms almost. You can see the structure of the proteins themselves. You can see the structure of the proteins so well that you can actually look at the amino acid sequence of the proteins. And you can see that the amino acid sequence is exactly what the genome would predict, the genome that was measured. So in this paper, what is this that they're looking at? What is this that they're measuring? They have protein experiments here too.
Yeah, so Dan, a virus is not just supposed to be a protein. There's supposed to be an intact genome surrounded by the spiky protein shell.
That's what they're showing here.
So you're showing a protein analysis, but there's nothing in this paper that establishes that there's an intact genome, and it certainly doesn't establish that there's a genome surrounded by the spiky protein shell.
They confirm sequencing.
I know they did so-called sequencing, but this one they're working with 20 million reads from the Vero cell culture.
Yeah, they got millions of reads in the other one too.
Yeah, and they put it into, I mean, I can just read a little bit here. So from in this paper, they have the methods are in a separate PDF that people have to find online.
Which is standard for this journal.
So, yeah. So, sorry, I'm just trying to find next page. So they took the total RNA, and this would be coming from their centrifuged viral cell culture supernatant. They took, they say they isolated the mRNA from the RNA. They made complementary DNA. They did some amplification. They ended up with a library that they loaded into Illumina. They had 20 million reads. They weeded out reads that corresponded to the monkey cells, the Vero cells, and they did some trimming, and they fed this into a, quote, guided assembly galaxy pipeline using default parameters, digital normalization, and a reference genome to guide the assembly of the genome. Because they don't find an intact genome, again, they're assembling a genome.
Which is how sequencing works. If you're going to discount this, you're going to have to discount all sequencing. You're going to have to discount all human genome sequencing. So you think human genome sequencing is invalid?
I haven't studied human genome sequencing. I don't know what they did, so I'm not going to comment on that. We're just here to discuss this paper.
Right.
So what I'm seeing is, again, they've got literally millions of reads, and it's being fed into an assembly pipeline.
It's just a computer algorithm that is doing what scientists did by hand for decades very quickly. You could get the same results if you do this by hand. People have done that.
Dan, so they assembled an in silico genome. They haven't found an intact genome.
So how was the genome? Do you know how the genome was measured?
There is no genome. That's my position. They have millions of individual reads.
How are those
reads obtained? Into an assembling pipeline.
How are those reads obtained?
Well, we just went through it. They took the total RNA from the viral cell
No,
supernatant, and then
how
they
are the
made,
reads obtained? How are the reads obtained?
I don't know exactly how they come up with them. They find a bunch of sequences and then
Well,
they feed them into a pipeline and it assembles them.
so this is the problem,
Like
Christine.
obviously,
I showed you earlier how it's done. I showed you how
I
it's
think
done.
we can, we obviously,
It's done in a chemical reaction. They measure the sequence. And
I think we can just leave
and
it
the odds
there.
of the sequence, the odds of the reads overlapping the way they do are astronomically high, too high for it to be random.
Dan, I want to mention one other thing about this paper. So this paper, it explains in the methods, they didn't start out with any clinical sample. It's not anything that's come from a patient in this paper.
Well, it has come from a patient.
And
So
they
I
accepted
know.
an isolate. They accepted an isolate from G. Dobler
And
at
where did
the
the isolate come from?
Bundsquare Institute.
Do you,
People can find that out on my website because I did some investigation and I emailed G. Dobler and his
I'm aware.
response is there on my website. So
I'm
people
aware.
can find out.
But do you know where the
It's a
isolate came
monkey
from?
cell mixture. It's another monkey cell mixture.
Before that,
And
where did
like
it come from?
it's a monkey cell mixture, Dan.
That's not true.
It's on my
Where
website.
did it
It,
come
people,
from?
it It's from a paper by Corman and Drosten. I can pull it up. I actually have the details here right now in front of me.
I can say it.
So people go to my,
The name of the strain is BavPat1. It stands for Bavarian Patient 1. It came
yes,
from a
and
patient.
it also goes by MUC-1 or
Yep.
MUC-IMB1.
Yep. It
And I
came from a
wrote
patient.
to G. Dobler and I had him confirm where it came from. And
Hmm.
people can go through the information I put on my website. If they just type in MUC-1, they will find this page. And it traces back to a paper by Corman and Drosten. And it's just another study where they did monkey cell culture and
There's nothing wrong with
they
that.
interpreted cytopathic effects in a cell culture as evidence of a virus, and they're calling this their isolated genome, and that is not purified particles. So again, we don't have anything that was shown to come from a patient, and we said we're only discussing what's in this paper. The reality is these authors
Hmm.
accepted a substance from somebody else, and their idea of verifying it, according to the paper, was they did Sanger sequencing of an S gene fragment
Didn't you say that you
of a
wanted to see Sanger
fragment.
sequencing?
I'm sorry.
Didn't you say that you wanted to see Sanger sequencing?
So Dan, they didn't sequence a genome. They're talking about an S gene fragment.
And do you think
This was
that
their idea
is that
of
S gene
verifying
is
a
that
full-length
hang on.
genome with a spiky protein
Hang
shell.
on, let's pause there. So Sanger sequencing of an S gene fragment.
Yeah.
You accept Sanger sequencing, correct? You established that earlier?
Dan,
Yes or
we're not,
no? Just
they're not,
please.
it doesn't matter, Dan. They're not talking about a
Okay.
full-length genome. They're
Okay.
talking about,
So
and there's
we'll
no,
say hang on. We'll say you accept Sanger sequencing. So that fragment that they sequenced, if you took that sequence and you compared it to all known sequences and you saw that it was unique only to SARS-CoV-2.
Dan, how do you know that the whole purpose of looking at these papers is to establish whether this SARS-CoV-2 is even shown to exist? You don't get to
Oh
assume
no, that's not the
that you
purpose of
found
this
a
paper.
sequence from SARS-CoV-2 when you're giving a proof of SARS-CoV-2.
Hang on, Christine,
You don't get to do
let
that.
me explain this again. So, nothing is assumed. You said earlier that you accept Sanger sequencing. They empirically measure a sequence using Sanger sequencing. You can take that sequence and compare it to all known sequences, and you can only get SARS-CoV-2. In that
Dan,
instance, where do you think that DNA is coming from? Where do you think that sequence is coming from?
I think we can just stop.
Christine, you
We're
haven't
not
answered.
going to get anywhere, Dan. I mean, again,
I'm trying to get somewhere with you, Christine, but it doesn't seem
yeah,
like you want to learn anything
I
or
know
answer
it.
any questions.
We don't see eye to eye. I see a paper where 20 million sequences were fed into a guided assembly pipeline.
And you don't know what that means.
They did not find an intact genome. They're making things up in a computer again.
No, that's not true. So let's look at the cryo-EM. Again, I started explaining that with this paper and you didn't answer my question. So with their cryo-EM results, what are they looking at?
Presumably a protein. I don't really care because they
So
can analyze a
where does the
protein all
protein
they
come from?
want.
Where
They
does the
haven't shown
protein
that
come
the
from?
protein forms a shell around an intact genome.
That's exactly what
And they
they show.
certainly don't know that there's a virus because
Figure 3b.
there's no animal experiments, and we don't even know that what any of this stuff that they're looking at was ever in a sick person or any person at all, period. They
Figure
accepted a
3b.
mystery substance, they put it into some viro cells, they
Yeah,
did some
I know. We went through that. We went through that
centrifugation.
already. We're not going to keep belaboring this stuff. But I'm going to
Yeah.
keep asking you questions because
We're not
you
here
are
for you to
Christine.
question me, Dan. We're here to discuss what's in these papers.
Yeah, and I can ask questions during a discussion. It's completely fine. But here, look. If someone is going to make a really impactful finding in a field or overthrow a view in that field, they should at least know things about that field, right? I'm not going to make a, a paradigm shift finding in quantum physics when I don't know what a muon or a gluon is. It's just not going to happen, right? Similarly, you're not going to overthrow virology when you don't even understand what you're looking at or what these methods are or answer simple questions like this. This cryo-EM is unambiguously showing a specific protein, and it's showing that protein embedded in a lipid bilayer that forms a shell around a coronavirus particle. Where does that protein come from?
Dan, nobody's disputing the existence of proteins.
So
You have a mystery
but
substance
where
that was
does
mixed
this
with vero
particular protein come
cells.
from? Where does
Who
this
knows?
particular protein come
It
from?
doesn't matter. It's not shown to come to be a shell around a full-length
So
genome, so we don't even need
you
to
don't know where
discuss
it
it any
comes from.
further.
You don't know where it comes
It's
from is the
coming from
answer.
a mystery substance that was then added to vero
How
cells, which
does
are monkey kidney
how do
cells.
proteins get made?
I don't know, Dan, and I really don't care.
You don't know how
You're not.
proteins get made.
No, I don't. I'm not a chemistry background. What I know is they accepted a
That's
mystery
that's basic.
substance that
It's
we're not
not
making any assumptions about,
Christine,
and they mixed it with
hang on,
vero
Christine,
cells, and then they
hang
found
on,
20 million sequences.
hang on,
I don't
Christine,
care about a protein because they can't show that there's an
hang
intact
on,
genome
hang
or
on,
that there's
Christine.
a protein
Just,
shell surrounding the genome.
just.
And they certainly
I
aren't
know.
showing that there's something
You
fitting
want
the
to keep saying your
definition of a
mantra. You want to
virus.
keep saying your mantra. I know. But
Well, you can call it
listen,
a mantra, but it's really just common sense.
it's really just your mantra. With
They
in the
didn't have
Christine.
purified particles, extract the RNA from those
Yeah.
purified particles, and find
I know.
a full-length genome.
I know what
20
you're gonna
million
say. I know what you're
sequences.
gonna say. But
Dan,
Christine, if you
I
don't
don't
know
mean
where
to be
proteins come
rude.
from,
I'm not a night person and I had a headache all day, and we're not going to agree. Like, we know we're not going to agree. God bless you. Thank you for having me. You've been polite. I'm really impressed that, we did as well as we have. I think
I
we're
think
making progress in
I
that
think it went about
regard, that
as
we've
well as
we've
expected.
been
So
nice to each other. So thank you for having me on. We're just going to keep going in circles, and there's no point. So thank you for having me. God bless you. Have a wonderful evening. I'm sure we'll cross paths again.
all right. Well,
Okay, is that all right?
sure, you can go if you want.
We've been an hour, and honestly, I'm not a night person, and we're just going to keep
That's fine.
talking,
I
like
understand.
going in circles.
I
So
understand, but please, when you get off this call and you wake up tomorrow, at least please look up how proteins are made. It's really important.
well, if someone could show me first a full-length genome, then I would consider delving more into proteins. But if there's no
You gotta
full-length
learn the
genome,
basics. You gotta learn the basics, and I promise DNA is involved in where proteins come from. So
20 million reads, Dan, and no animal experiment in this paper. It's just
those are in other papers that you
we're
didn't
just going
want
to
to
go in
discuss.
circles. So,
Those are in other papers you didn't want to discuss. So, okay, uh, I'll
okay.
let you go.
Thank you. Have a good night, Dan.
You
God
too,
bless.
Christine. Bye bye. Wowzer. All right, let's get this. Uh, no, it's not going to let me do that. Okay, cool. Um, let's get SenseStrand in here. Yeah, that went about as expected. Um, wow, I wasn't expecting her to actually say she doesn't know where proteins come from. That's pretty wild. That's pretty wild. Wow. Uh, let's see. Email. Yeah, yeah, yeah. One sec, man. Amazing. All right, let me see if I can actually. The chat has been like frozen on the little snippet that I can see right now. So I actually don't know what people have been saying, but hopefully it like refreshes or works at some point. Yeah, I can't see it. Oh, there it is. My bad. Whoops. I see. I'm just silly. Whoops. Jesus Christ. There we go. Hey man, how's it going?
I'm muted. There you go. Hey.
How's it going?
Oh, great. You had a fun time there, did you?
That was interesting.
That was fun.
Like I said, I did not expect her to say she doesn't know where proteins come from.
They're left at no last resort. She knows where proteins come from because she's been asked it before. She knows they come from cells and that those cells generate the proteins through specific sequences, which we call RNA, which come from DNA.
Mm-hmm.
It's just, you're stuck. Once you've admitted that step, that's the central dogma of cellular biology, then you have no place left to go.
Right.
And she had no place left to go. That's why she suddenly had a headache. I've been having a lot of sudden headaches. Oh, I'm actually really tired. I need to go to sleep now.
Yeah. Yeah, that was. Oh man, I need to get the. Let me see if I can get the chat up here.
Go ahead.
It's gonna take me.
Lots of fun people in the chat.
Oh really?
Alec was in the chat. Mike was in the chat. They're welcome to come up if they have.
Mikey Stone.
Oh yeah, if they have any capacity to do so.
Oh yeah. They're welcome. Yeah, it's gonna take me. I'd have to like reconfigure my settings.
No.
So
Yeah.
we'll just go.
I'm watching I can watch the chat through the thing. Do you want to take questions from the chat?
Yeah, I can start taking questions. Let me get on my phone and do that.
Unfortunately, I think most of the virus deniers basically have run away.
Well, that tracks.
Yep. It was quite a train wreck for her. I will say, you brought in the nanopore paper. The nanopore paper did it all in one shot. So I've not gotten an intact genome out of this.
Yeah, I said that to her, that this original isolation paper uses nanopore sequencing, and that wasn't enough for her. She just kept saying her mantra of it's not an intact genome, blah blah blah. Just wanted to say that over and over again.
The thing with the 20 million reads is it's not a random bag of reads that you pull out and then assemble like their Lego blocks that can just fit together anywhere.
Right.
They will only fit together where there's overlap because they come from one sequence.
Exactly.
Based on homology.
Yeah, and she didn't seem to understand the overlap part. She thought 3 nucleotides here, and that's not how it works. Reads overlap by dozens of nucleotides, if not hundreds, when all is said and done. And
then those overlaps interlock in, and they have other overlaps to them. So they're not So
Right.
it's a full genome. So once you run out the mathematics, so she says, well, it's just a probability. Is like, yeah, those probabilities are beyond astronomical once you've added in. And I run this calculation with my students every single semester. I say,
Right. So
why
like,
does a why does a 20 base pair primer, how can it go and match one specific spot in a billion base pair genome?
hmm
It can do that because every sequence adds on to each other, right? So you have 1 in 4 chances of ATGC, 1 in 4 chances of ATCC. So by the end of 20 base pairs in a primer, it's 1/20 times 10 to the fourth.
yep, 1/20 times 10 to the fourth. Yeah, that's a
Yeah,
lot.
so the chances of that are astronomical. That's
Yeah.
that's why a primer can match like that.
Yep, and that's just
And in terms of metagenomic analysis, is you can. He's like, well, it's a soup. You haven't isolated it. It's like, well, we can do much more complicated things than that. You
right.
could take a cubic inch of soil, right, which has hundreds of thousands, if not millions of individuals, but hundreds of thousands of different species in a clade, and we can align those up into their individual clades and identify them.
Yep.
So
Yeah, that's
identifying one virus is is nothing.
yeah, that's what you brought up with Koreshi, where you
Yes.
asked him about putting like human and plant DNA in a blender, mixing it up, and asking if you
Yep.
could figure out
Parsing
which
out.
which DNA was human and which was plant. He said yes, and then realized he messed up and then just had a headache himself.
Yeah, rage quit.
Yeah.
A lot of rage quitting.
So you
So,
know
do
what?
I,
Let
would
me
you
just
recommend a second bivalent shot?
do this.
Go
Let
ahead.
me. I'm just gonna put this on screen, um, so that I can see this. The stream is kind of frozen, so you can't really see that. But anyway, now I can see the chat.
Okay.
Um, Sophic Manatee, thank you for the super chat. I always say you guys don't have to do that, but I very much appreciate it. Um, wow. Okay.
Suffolk Manatee, sorry for giving you like a little bit of shit. If I can curse, sorry.
Oh
Uh,
yeah, fine.
he posted he posted a I was on my other account. He posted a picture of a skeleton, and I said, because he was waiting for the evidence, and I said, was that guy on his fifth shot? And he goes, no, that guy was pure, no shots.
Wait, who? Oh, you mean the skeleton?
Yeah, yeah, yeah.
Okay, okay, I get it. Do I recommend a second bivalent shot? I mean,
Second bivalent shot.
a
No,
second one. I don't know. I don't think so.
no, that's not
No,
what's recommended.
no.
Unless there's another variant that changes immensely.
Yeah, I mean the situation with vaccines right now is like the only people who should be getting more boosters are the ones who are at extra risk, like extra
Yeah.
high risk, because it's not going to do much more for you. Um, Getting your 3 doses, you're good. Most people. 2 doses are even good for some people, but if you want that extra protection that is going to broaden your immunity and make you better prepared for whatever future variants might come your way, 3 doses is best. Federico's here. Hey, what's up?
Yep. Yeah, Federico's here.
Yeah, sorry.
Uh,
I'm just seeing the chat for the first time tonight, guys. I need to get a third monitor, is what I need to do. I finally got like a cool little background I found, and like
looks nice.
I figured out how to like configure my screen. So I said that eventually I'll get better at streaming, and I got a little bit better today. Um, Jubei looks pretty mad.
Yep,
Um,
Jubei is pissed off
yeah.
for sure.
Uh,
And they
let's.
keep calling it a computer generated sequence. If it's computer generated, I will eat my hat. If you can go into any data set, any data set, prior to 2019, and generate me a SARS-CoV-2 genome. Use
Yeah.
any pipeline, anything you want. I mean, we could put that up as a full-on like escrolled $10,000 challenge. I would do that.
Yeah. Yeah. You could, because it's not going to happen. Like, no matter what you like, I don't think. I think what's hard for people like Christine to understand, or maybe just admit, is that there's physical, like, chemistry that you do before you ask a computer algorithm to analyze the data for you. Like, it's not just all done in a computer. You have to actually physically obtain data. If you just generate random sequences that you feed into an algorithm, it's going to give you nothing.
Right.
But if you
No.
actually measure a genome, like what they've done, what so many scientists have done all over the world and found the same thing, then it's not computer generated.
I think showing how you can tell when this is C. Kai. I think you can show how telling how sequences are not part of the same genome in Illumina sequencing would clear up a lot of this up for her and her followers. They hinge their arguments on this, but that would take a lot of. I mean, we could probably bring up an alignment software and show that the alignments don't work, but would they believe it? That's too much.
Yeah.
They won't understand it.
Yeah, I mean, they have to get the basics to I think the problem, they're not going to understand next gen sequencing if they don't understand the basics first. Like, if you can't get past the fact that if you can't get past the principles on which Illumina sequencing is based on, and
Yeah.
accept those as a valid way to sequence, then you're not going to get anywhere.
No, you're not going to understand alignment. You're not going to understand homology. Okay, let's see.
But yeah, the point that I was trying to, like,
Some nonsense.
get with her at the end is, like, I think that I really can't drive home the power of cryo-EM enough in these settings, because I mean, seeing things on atomic resolution is just insane. Like, I know that some virus deniers, maybe they're in the chat, will say that this is just a computer generated image, but it's
Not.
it's not that's not correct, because what they're doing is they're getting electron densities, which some of them are shown here.
You might want to share your screen.
Is it on the stream?
Oh, hold on.
Should be on the stream.
No, I don't. Guys in the chat, can you let us know if you can see it on the stream? Oh, hold on. One sec. I gotcha. Okay. Sorry.
Okay.
I just realized that
There might be a delay. But yeah,
there is, I think, a second camera going on the stream that I can see.
second camera. What is that?
Yeah, like instead of me, it's another picture, but not with you.
Really?
No, that's okay. It's fine. People can hear us.
That's
You
weird.
don't have to see my face.
On my screen, it looks like it should be working. But anyway,
Unfortunately, I don't know if you can see right up in that corner there.
oh, that's weird.
Yeah, that's weird.
Why is it doing that?
Probably gotta refocus. Whatever. People can hear me. There you go.
Did it
Oh,
work?
yep. Now it worked.
I barely did anything. Okay. Anyway, yeah,
Yeah,
these are
moves. I don't know.
these are electron density gradient. Electron density maps. So you're physically
Yep.
using an electron microscope to shoot electrons at these molecules. The electrons bounce off the molecules and hit detectors. And the angle at which they hit detectors allows them to use calculus to back calculate where that electron was deflected off of its
Right.
forward path. And so by doing this with thousands, tens of thousands, or even hundreds of thousands of individual particles, they're able to average those maps together, and get more and more confident about more and more precise locations of those atoms that made the electron bounce off. And
Right.
so then you end up with this electron density map, and you can actually fit specific amino acids to those densities. And so that's how you can see the sequence of the protein. And this protein has to come from somewhere. That's what I was trying to get her to understand. It
Yeah.
has to come from DNA, which is transcribed into RNA, or in the case of the virus, it's just directly translated. But DNA, RNA, protein. So some genes had to make this protein. And where were those genes? Those genes
Where
aren't
do they
in,
go?
those genes aren't in humans. Those genes aren't in monkeys.
Right.
The only gene that can make this protein you find on a coronavirus. So
Exactly. Exactly. They have no answer for it. And
yeah, there's
I mean,
no
how
answer.
many times have I gone back and forth with that with Mike? Is, well, I don't want to talk about proteins or nucleotides until you've narrowly defined what I expect.
Oh yeah, you got your kid in the background. Um, all right, let me look at chat a little more. Jubei's gone, I guess. Oh no, he's still there. I don't know if I have any mods in the chat tonight, but
I'm on. Sorry.
yeah, I don't think they're putting people in timeout. Um,
Ooh,
let's see.
painful
Any
watch.
any other questions or things that I missed while I was on with Christine? Mash your caps lock like Jubei.
Yeah. No, dude, they found a fragment of an S protein. She's right. It doesn't matter how proteins are made. You can make fun of her all you want. Okay, runners, meditation.
I
Where
see.
does the protein come from? How was it made? Why do we detect it?
Yeah. What do you mean it doesn't matter how proteins are made? It's the whole thing. Like it has to come from
Guys,
somewhere. So where does it come from?
it's the basis of all of biology. So,
Yeah. The thing about I had to start saying that she was just saying a mantra, because she really was just repeating it over and over again, the exact same thing. No matter what question I
right.
asked her, no matter what concept I tried to get her to explain the way she understands it, she just went back to her mantra of the script that she wrote out.
Oh, yeah.
And it's bizarre. It's bizarre how, like, you know, I'm trying to have a conversation with someone, and I ask them a question, and they just will not answer. Like
Nope.
it's a trend with these virus deniers. They just will not answer questions.
Nope. No, they want to argue in their specific way. And then once you've asked and answered their question. So a couple of things. I was communicating with Bitcoin Bum, and her entire thing was, sometimes they will have mock, and the mock won't include the first round where they're adding in the stronger antibiotics, right? So then I said, okay, let me provide you with the TCID50 test, which, by the way, and have you checked out covidresearch.net yet?
Sorry, COVID. Oh,
Covidresearch.net. It's our
wait, the database, or the
database.
what do you call it? No, sorry, I haven't gotten to look at
Yeah,
it,
cool.
but
It's everyone should check it out. It's our
oh yeah,
thanks Independent Mind and Federico put it together, and I helped them assemble it.
yeah,
And
I need
basically, we
to.
have about 900 papers with notes, so
Hmm.
it's pretty nice.
Yeah, I need
Uh,
to see if I can add to it.
oh yeah, there's a lot of things that are still missing.
Yeah.
But then, so one of the things that we're back and forth about this, I provided them a TC 50, which they refuse to acknowledge. And they, and all this is actually thanks to K-State. Our buddy K-State was the one that pointed out the TC 50 because it's just a standard 96 well plate, and then the dilution assay across. So if the control treatments doesn't work, then how come the dilution causes less CPE?
Right.
That you can see going across the plate.
Yeah, so I think I have on screen here an example of TCID 50, and yeah, this is exactly what it looks like.
Yeah.
So I actually do this test a lot. We, funnily enough, use a virus that does not produce plaques, so we can't use CPE to define a positive. We actually use qPCR. So we measure how much virus is actually replicating in each well to determine its infectivity.
Right.
So, but yeah, this is a TCID 50. You have your serial dilution, so you have your virus or your sample, which may or may not have a virus. You dilute it across multiple tubes, 1 to 10 dilutions, plate those all the way down the plate. You have replicates going across horizontally, and the dilutions are going vertically. And as the dilutions get more and more dilute, so as the virus gets more and more diluted out, the purple, which I guess is presence of purple, is healthy cells. This is probably like some sort of
it's a crystal blue,
crystal violet stain.
Violet.
I could look through it, but it doesn't matter. The more purple means more live cells. So as you go down in dilution, there are more and more live cells. And the control, look at that. All those cells are healthy.
Yep.
And when the virus is most concentrated, least diluted, all these cells are dying. So
so how is that possible if it's not? And then they'll use a really nice paper. And actually, can you just go to covidresearch.net?
Yeah. covidresearch.net. There it is. So tarot.
Okay. Yep. So we have things broken down by arguments, virus deniers, etc. If you go to the virus deniers argument one.
Yeah, let me just link this in the chat real quick. I know probably Federico might have posted it already, but I'll post it.
And most of those are posted by Federico.
Go to what now?
Virus deniers arguments. There you go.
Yeah. Cryo-EM. Oh, I can add more to there.
Yeah.
Direct.
Yeah, the cryo-EM needs more. If you go to the PCR, do I have the PCR one?
PCR.
They also quite weak.
Oh, okay. Yeah, I could definitely add stuff.
But I only go to that one paper I have. I might have two papers. The first paper that's in there.
The PCR one?
Mm-hmm. It's the PCR times the cultures, right? Where they took like 6,000 or 4,000 samples of people who had COVID symptoms, and then they ran PCR for week 1, 2, and 3. And then showed the culturing sample ability, right? So they ran it on a culture sample.
Oh, okay. Oh, they used a plus minus culture assay, right?
Yes,
okay.
They did. But basically they were saying, so you have two independent assays that show, okay, one's testing nucleotide abundance with the PCR assay, and the other is showing the ability of the virus to be a live virus to infect cells and cause CPE, right? So they're correlated. The higher the amount, the higher the CT values, right? The longer it is to, and if you click on it, it actually will bring up notes and a picture of of the thing. It's like, so they're using it as like, look, PCR doesn't work because of all these false positive. It's not a false positive, right? It was a true positive because the person got sick, and it was exactly correlated with how the higher CTs led to lower cultures being positive for the virus. So that's an interesting trick. How do you do that? How do you pull off two independent analyses coming to the same conclusion?
Right.
If the CT values were 25, then 90 to 80 to 90% of the cultures that they tried to use had CPE. So how do you if something doesn't exist, how do you get two assays to agree independently with each other?
Yeah. Yeah.
And then when they tell you, well, if you have healthy human, if you went out and got a healthy human just randomly off the streets, right? It's the the control was invalid because they expect the control to be you picked a random healthy human off the streets and got their sample and showed that the tissue sample isn't just producing CPE because of some random human that's not sick. And now, okay, now you have healthy humans because they're no longer sick and they don't produce cultures, CPE.
Yep.
So,
Yep. Yep. There's not enough virus there to produce culture or produce CPE. Yeah. Yeah.
and in fact,
This is what they want. I mean, that's
on a
yeah.
quantitative level,
Yep. Yeah. Like on because yeah, for those who might not know, on Twitter, there's been a lot of virus deniers just saying things, and this is kind of what they say. They want to see. They say no, a mock control is not good enough. I want a sample from a healthy person, and there you go. I tell them, well, that's called a negative sample. It's
yeah,
just it happens all the time. You test
right,
you test you test a patient for specific pathogens and you get negative results.
right.
You test an asymptomatic person for pathogens and you get negative result.
To the group
Yeah,
is
people.
there's-
Just another thing, people often use this paper to support the misinformed idea that PCR
There you go.
doesn't work. I've been through that before, so we don't have to belabor it. But
It's a really good paper. It shows
yeah.
very specifically how accurate PCR is, right? And they're like, well, it was positive. Yeah, at 35 cycles.
Yeah, and oops, and I might as well just explain now. But yeah, even if it's positive at 35 cycles but can't infect a cell culture, that doesn't mean it was a false positive.
It's a false positive because it's a matter of it's a waiting game, how much exposure, right? So you have
Yeah.
one sample, you go in, you get one sample, and then you put it on a plate. That's like one time point. But if somebody's sitting and breathing on you on a plane, right? That person might have been sitting and breathing on you for 3 hours, right? What's the flow on that?
Right. Right.
Right. So anyway, we could go back to the
Sorry,
chat.
I hear my
Go ahead.
I hear my daughter crying.
Oh, you want me to try to take over?
That's not good. She never cries.
What baby never cries? Oh, she's a toddler now, isn't she?
Well, she cries. I shouldn't say that, but she never cries after we put her down. She usually sleeps really good at night, and I hear her making noise. Hopefully she'll quiet down. She'll go back to sleep.
Show me photosynthesis. I don't know.
Maybe I'll
Just a bunch of stuff. Still Jewy, insults, logical fallacies, ignorance. This is what they'll do. They'll just shout logical fallacies without saying what they are or having them make sense. Alec Zek's favorite one is, oh, well, you're just begging the question. It's like, no, we're not. The test is specific. We ask the question, and the test answers it for us. We
Right.
have inductive reasoning, not deductive reasoning.
Right. Right. I mean, these are the same people. I mean, Tom Cowan has questioned whether ribosomes exist. So apparently, all of molecular biology is fake. Jubei says prove viruses exist and cause disease, but you can't. Yeah, we can. So
Yep.
tonight, Christine only wanted to discuss two papers, and so I told her we could open the discussion to the literature, and I can show you like any experiment you want to see to demonstrate any part of virus causing disease. And she said no. So we
Did
eventually,
you
after like
did you
50,
notice she immediately broke her rule where it was you were only supposed to talk within those boundaries of those papers?
oh yeah, yeah.
Once she was cornered, she ran to uh, Lanka.
Yeah, yep. Nope, Blanca did not do a proper control experiment. But anyway, like um, there are plenty of papers that show that you can take a SARS-CoV-2 isolate and use it to infect animals. So they've done it with non-human primates. They've done it with mice, but the mice specifically had to be modified so that they could be susceptible to SARS-CoV-2. If mice,
Right.
because mice don't express ACE2, which is what the spike protein binds to. Um, and so SARS-CoV-2 is not going to infect them and make them sick unless they're expressing ACE2. So you can take mice, get them to express ACE2, and then get them to be sick. But if they're not expressing ACE2, they won't get sick. So we have animal studies, and I see some virus deniers on Twitter saying like, oh, I want to be able to observe it in nature. Well, we can do that too.
Yes.
It's called contact tracing. You can,
Right.
but of course you have to believe in sequencing and stuff to get that. But you can essentially watch you can watch a virus spread from person to person throughout a community, and through contact tracings, through sequencing the virus from every single person who tests positive, you can come up with a map of who the virus infected first, who it jumped to next, and then next, and
Right.
then next. You can observe it in real time. So contact tracing is seeing virus transmission in nature, and you can watch those people get sick.
Right. And that's how you would. So it's not like we can't detect it in nature. The problem is it's a false argument that what they're going to do now is like, well, no, but the way that you tested it is you brought it into the lab, right? So it's like, yeah, no, there's no electron microscope in nature. There's no PCR machine in nature, right? And because these things are submicroscopic,
Hmm.
you can't visualize them in nature. You have to use the tools that are in the laboratory to see them.
Hmm.
That doesn't mean that it didn't come from nature.
Yeah, it's the flat Earth argument of, oh, you're using a gyroscope. That's not natural. That's not a scientific experiment. It's like, what? What are you talking about? They
Did
just don't.
you? I don't know if you saw the thread, but I tagged it, and it's virus deniers fighting with flat earthers. That happened today.
Oh, it happened today. Oh, I
Yes.
didn't see that.
Oh, I have to show it to you. Um, Basically, they just admit it's like we have our logic is strong, and it basically mirrors flat Earth, but we shouldn't go there. Is what they say specifically.
Oh man.
I got it. Actually, if you go on your Twitter, search hashtag VD bad takes.
Okay. Let's see. We can do this, and then I'm gonna wind it down soon because I want to make
Perfect.
sure that my daughter didn't. Uh, my daughter went back to sleep because she almost never cries um, when after we put her down, but in the past, when she has, uh, it's been because she's gotten sick. So hopefully that didn't happen VD bad takes. Here we go. Oh God.
Should be the top one.
Top. Um,
You didn't see it?
latest. Is it by you?
Yeah. No,
Who posted it?
that would be. Yeah. Here we go. I'll tell you about that later. I'll just send it to you real quick if you can't find it. I see that it's for some reason not giving you the latest one.
Who is?
Oh, it's there. It's there. It was on time. Go to the latest, and I got a delay on my screen. That's why I
Yeah.
can't see.
Yeah. Oh wait. Oh, okay. Here it is. I
Yeah.
appreciate the extreme logic used
The
to
extreme
know.
logic.
Okay. Wow. To know that viruses don't exist, but with some it goes too far into flat Earth theories. Extremity in any form leads to detachment from reality, including logic. Can I prove equivocally that the sun is a sphere? No. Do I know it is? Yeah. What? What?
So a tacit admission that
Wow.
their version of logic, they can't take it too far because it just makes them flat earthers.
Wow. That's I mean, they're getting closer to admitting that they're a flat earthers of biology. Does the virus believe in me though? Viruses believe in you.
They probably don't want.
They
Sometimes
they need you.
I was I think I hijacked one of Luther's spaces that he didn't let me on. Now he's let me back on, I guess. But
Oh,
I
he
told him
he,
I'm
he
not
unblocked
coming.
you.
Yeah, he unblocked me. And one of the people were coming up that had to bring up, like, he was so enthralled with cordyceps mushrooms and how they can infect ants and make them into zombies,
Mm-hmm.
but could not for the life of him just say, like, okay, well, what are that humans also get pathogens, right?
Mm-hmm.
um, but it made me wonder if the virus could infect us and change our behavior to ignore it.
That would be something.
Yeah.
This person actually I wanted to start with this, but I thought it might be too rude and make Christine leave. But oops. Um, they ask, Dr. Wilson, I understand you want to tackle this issue, but when people don't believe in viruses, there's no evidence you can show them that'll convince them, so why waste your time on this topic? So my goal going into this was not to convince Christine. I know that she's not going to change her mind. I don't think that she is mentally well. My goal here was to have someone who has influence within the virus denying community, and she has a lot of influence in that community, to have a conversation and show to the viewer that they don't know what they're talking about. That was the point of having this conversation, because hopefully in the future, when people are susceptible to this idea, that when they hear the idea, oh, maybe viruses don't exist, and they think, oh, that might be true, maybe I want to look into this. Hopefully they Google, they find my content, or find something
Right.
like this, find something like this, and watch that instead of just Andrew Kaufman or just a Cowan video. Because when it's just their content out on its own, then when people go searching for this topic, then that's all they'll find, and they might slip deeper into the rabbit hole. But at
It's in a
least if there's
vacuum.
something
There's
there to
nobody
challenge it,
challenging them.
yeah, at
Yeah.
least if there's something there to challenge it, then maybe those people might not slip down into that rabbit hole. So that's the point here.
Yep. I think it's a worthy point. That's why we push them on it.
Yeah. So hopefully this helps someone who might be susceptible to virus denying beliefs.
Right. it's just sad because they will never progress. Next year, in my own science, which has nothing to do with COVID, I will progress and learn something that I did not know last year.
Hmm.
And the year after that, I will know more. And the year after that, I will know even more. And those each of those experiments are designed to test the previous knowledge and answer new questions. And
Hmm.
that's how virology works. But that's not the only thing that it's not. They seem to think that virology operates in a corner by itself,
Hmm.
and that there aren't protein chemists and metabolomics peoples and genomics people and cellular biologists who are just ignoring what virology is doing and saying like, oh well, they're doing their own thing. It's like if virology didn't work,
Yeah.
then none of cellular biology would work.
Right. Right. That's the whole thing that I was starting to try to explain to her when she said you're assuming, and I said no, it's testing. Like these assays are designed to ask the question like what is in this sample, or is there a virus in this sample, and you can get the answer of no. Like
Yeah,
it's not assuming, it's asking a question and then having a test to answer that question, whether it's yes or no. And then if you design other tests to build off of that, then those tests won't work unless the original result is what you think it is.
yeah, yeah. And you have the ability to kick somebody out of the comments, right? The
I
live feed.
do. I do.
Okay. Can you go ahead and kick out Cordelius too?
Oh,
He
is
just threatened physical violence on somebody.
oh really? Where did
Mm-hmm.
oh he's challenging
It
someone to
was,
a fight? Wow, that's
yeah,
you're in timeout, buddy.
yeah.
God, these people
Boom.
when they who was it? Someone on Twitter, I think, in the past month wanted me to meet them in person. I can't remember who it was, but I remember someone doing that. And it's
Oh,
always just an it's always just like an admission of defeat when someone does that.
basically.
Oh
Yeah.
well, you embarrassed me online, so now I want to punch you.
Right. Exactly. Well, maybe you shouldn't just get uh, so dumb. But it's at the point that they're at. I don't know. Whatever. It doesn't matter. Like I said, the concept of viruses will continue to evolve. There was 50,000, 55,000 publications, scientific publications alone for viruses uh, last year. Next year there'll be 60, right? The
Yeah,
year after that there'll be 70. That trend just goes upwards because people are learning more and understanding more. And in 10 years, Mike and Alec or anyone else who's still in the virus denying movement will still have the same arguments they had 10 years ago.
they'll be saying the same stuff. Yep, they'll have learned nothing.
Right.
That's what's
So,
interesting to me. Like, I mean, when I was, like when I was deep in conspiracy theories, it was like the same stuff over and over again. And like I would try to find new things. I think that's why conspiracy theorists often jump to other believing other conspiracies because there's only so much
right.
they can explore within their own conspiracy theory because
There's nowhere to go.
nothing's ever going to change.
No.
So they either like have to evolve or just like jump to other conspiracies. Um, but that's what they're going to be doing 10 years from now, still saying the same stuff of there's not an assembled genome and you're assuming and blah, blah, blah.
Oh, so John Snyder 2006 asked, so did the flat earthers win? They did. In that thread, the flat earthers won.
Hang on.
Because the logic just goes to that address. There's nowhere else you can go with it. If you have that strong of a denial and you think everything is made up, then the earth is also flat.
Oh man. Okay, here's a flat earther. Maybe one day you'll see it. I used to hold the same view. Significantly, the theoretical curvature of the earth is not observable. Congrats on figuring out the virus scam, though. Most of the world is a deception.
Yep.
Oh man. It's like in Godzilla when he's like, let them fight. Except instead of Godzilla and King Kong, it's
It's
just it's like
BDs and
two
Flurfs.
ants, two like grasshoppers fighting. Wow, what a weird thread that must be.
Oh, it's something special.
Um, I saw I saw Demetrio came back.
Yeah.
Dr. Wilson, have you thought about contacting Joe Rogan to get on his podcast? There's no way that's happening, man. I'm like
No.
nobody on the internet,
No.
and Joe Rogan has like people booked for months to go on his podcast. He's not gonna have me on. Um, it would be nice to go on something like that, but I mean, if I even had the opportunity, I might just suggest someone else because I think it might be
No, he's
more
just not into
valuable.
conspiracy debunking anymore like he used to be when he ran
Right.
Joe Rogan that questions everything.
Um, Emlyn asks, can you do a debunking of HIV denier Peter Duesberg? I cover him a lot uh, in my review of RFK Jr.'s book because RFK Jr. talks about Duesberg a lot, and so I end up going through the story of
Right.
Duesberg and how he's a horrible idiot who denies that HIV even exists and that HIV causes AIDS. And he ended up contributing to causing lots of damage, not just in the U.S. but especially in Southern Africa, where he's kind of helped spread that belief and help it catch on. And antivirals were antivirals were not as used as they should be in Southern Africa, and even to this day, it's hard to. I mean, that region of the world is very heavily afflicted with AIDS, because people thought that HIV didn't exist and didn't cause AIDS, and that the antiretrovirals were poison. So, but yeah, I do go over that in my review of RFK Jr.'s book. I think it's chapter 4.
Yep.
He interviewed Kevin Folta a few years ago. Really?
Yep, he
Joe
did.
Rogan interviewed Kevin Folta. I didn't know that.
Yeah, it was a good interview.
Oh, that's awesome.
Thank you, Sith Wolf, for which one of the points? I just lost it. Shoot.
Sith Wolf.
Sith Wolf. Sith Wolf. Actually, a great point. Plenty of videos showing the cordyceps infection of insects. If insects get pathogens, why not humans? Yeah, I mean, you could just point out logical inconsistencies across the board. Parasites, they acknowledge parasites.
Oh yeah, anti-vaxxers and pe- conspiracy theorists love parasites or love trying to treat parasites.
Yep.
I was listening to
Nope. Straight up make up parasites.
Real Truthers Space earlier today while I was at work, and they were talking about it, how like especially in the anti-vax autism community, even beyond
Oh yeah.
that, parents are seem to like to try to treat autism out of their kids, or and the
Yep.
ways they do it are just horrible. Like it's child
Yeah.
abuse. It's just
Vital.
straight up child abuse because they think that parasites are causing autism in their kids, and so they like do like bleach enemas and just crazy shit to try and get the parasites out, and it's horrible.
Speaking
Um,
of along those lines, I didn't know that. What's his name? The one that started the autism vaccine,
oh, Wakefield.
Wakefield. I didn't know that Wakefield was that terrible of an individual. I thought he was just a data analyst. I didn't know that he was going through and doing all sorts of terrible things,
Yeah, no, he.
like like surgery. Elected procedures with um, colonoscopies on children that were totally unnecessary. And holy cow!
Yeah, yeah. Wakefield is awful. He is just a terrible, terrible person. Um,
Like, that's straight up child abuse.
yeah, and I mean, he lied to children and their parents. He lied about his results, all to try to promote his own MMR vaccine, which is pretty much what Robert Malone's doing today. Um, he's a modern day Andrew Wakefield, um,
Yeah.
except he's not. He
Well,
doesn't
he's not
actually.
experimenting on children.
Except he's not certified to actually treat patients, so he never got
Right.
to. He never got to get to the part where he lies to patients and collects samples from them in unethical ways. Um, but he's pretty much the same guy, same motives. All right.
Yeah. Do these chats stay on here? I think
Yeah,
they do.
they can
Okay.
you can watch them in a replay. Um,
Good. Because we
yeah,
have we have Alec coming in making some just really uneducated remarks
I don't
that I
think
would
I've
love to
interacted. I don't think I've actually interacted with Alec. But he's one of those
uh
people who has like he's one of those virus deniers who has like tens of thousands of followers on Twitter, right?
yep.
Okay. Yeah, that's the other thing. Just going back to
He's
that
a
question from earlier, like there are a lot of there are a surprising number of people who think that viruses don't exist.
yep. Yeah, there sure are. Uh
Sith
but that's
Wolf
why we're here.
Sith Wolf says, "Yeah, virus deniers have a trick for that. They deny cells. What about DNA? They deny that as well. Pretty much deny reality at this point." Yeah, I mean, that's like what I was saying to Christine. Like, someone breaks into
Right.
your house, um, which I got that analogy from Conspiracy Cats, by the way. I thought that was a good analogy. Um, but yeah, like someone breaks into your house, leaves their DNA. Someone breaks into another, same person breaks into another house, leaves their DNA. Like, do you trust that analysis when they find him and link both DNA samples to him? Like, do you trust that?
Right.
And she can't, she just would not answer. Like, it's a very simple
No, no, she
question.
floundered. She
She's
floundered on
not
that point.
honest about learning.
But
How
you had her cornered when she was like, "Well, proteins are real and they exist, and we admit that they do." Once she had said that, it became our last VD person that came on. It was just she had to shut it down, had to have a headache, had to get off because she said something that she could not possibly recover from.
mm-hmm.
That proteins are real, they're specific, they come from somewhere.
Mm-hmm.
Where do they come from?
Right, right. Yep. I mean, ironically, it's the same. I remember from way back when I first started interacting with Christine on Twitter. It was the same thing that made her like stop talking to me. Was I was asking her, I was asking her like, where does this sequence come from? Can you match it to anything other than SARS-2? Um, and she just wouldn't answer, and then stopped responding to me. And so I stopped responding to her. Um, how do virus deniers explain rabies? Yeah, toxins. Someone already said it. They literally think that rabies is explained from bacteria, mystery bacteria. They don't say what bacteria that produce toxins, mystery toxins. They can't identify the bacteria or the toxins, but that's what they say causes rabies. So
Lint Hamster Feld says clearly you get proteins from Amazon.
you can, yeah, yeah, yeah. I order,
Each cell has an individual order for proteins that they
yeah.
get from Amazon.
I order proteins from Amazon and I put them in my yogurt every morning.
That is also true.
That's not true. I don't take
That
protein
comes from hospital.
supplements. I'm a dad. I don't have time to do protein supplements and go to the gym.
Oh, you will at some point. Well, you will until number 2 comes along, and then you won't again.
Oh boy, yeah.
Yeah.
It's gonna be a boy, by the way. We know
Oh,
that.
congratulations!
Thank you.
Wow,
Yeah.
that's exciting.
Yeah, we didn't want
Yeah,
to
we're
wait. We just got the test, which apparently that test is DNA based, but I guess actually we don't know. It could be a girl for because it's a fake it's a fake DNA test.
that's right. That's what Copeland says. That's what Dr. Copeland says all the time too. Is like just throw all that out.
Yeah.
Go raw dog whoever you want because the baby daddy, you're not going to figure out who the baby daddy and mommy is. Um, don't worry about viral infections and STDs. All that's fake.
Yep. Yep.
It's like go really test out your theory.
Mm-hmm.
Go commit crimes. Spit and put blood all over the crime scene. You'll be fine.
I
You
have
haven't
isolated the cancel culture. That's good. Thank you all in chat for the congratulations. Yeah, we're pretty excited about having a second one. Um, it'll it,
Oh,
it's
yeah.
due. We're due in October to have the second one. So
Wow.
until then, my schedule for the schedule that I set for myself for YouTube will probably remain consistent. Once the second one comes, I don't know, man. I might have to like uh, put a pause on it for a little bit because it's gonna be rough.
Yeah, I would say so. Hopefully you can get
Um,
some extra
do I have
grandparent
any
support.
do I have any upcoming guests? I want to have more guests. I had Vicky Mail on, and I thought that was great. And having a guest is actually less work for me because then I let the guest do a lot of the like talking and bringing up sources, and then the editing together and putting the sources on the screen I can do like whenever. So it's ultimately less work, but I don't know who else. There are people I want to invite. You'll just have to wait and see who ends up coming on. I'm the limiting factor. I just have to like reach out and schedule something. But there are definitely people who I want to bring on who I know would be interested. Um, yeah, thank you.
Uh, in Twitter, one of the virus deniers took immediate issue with us calling them flat earthers, and a flat earther, Flat Mac, came on and rebutted him immediately. They're eating their own tail.
Have Dr. Wreck and Yellow on.
Oh,
I wanted to email him today, but I didn't get around to it. I do want to ask him to be a guest because there's a topic that I think would be topical to have him do sooner rather than later. So we'll see if I have the energy tomorrow to reach out.
yeah, Forrest Valakai would be a really good one. It's funny that Forrest Valakai and Professor Dave, and Simon Dan did one virus denier, but they don't go in the virus denier circles.
Well, you know what happens is often when Simon Dan, because I am subscribed to him, when he talks about like masks or vaccines, his videos get taken down. YouTube strikes them down. It's really weird. I don't know what I don't know what
Yeah.
this algorithm is about, but like my videos very rarely get flagged by the YouTube algorithm. But
Probably got to
man,
get like
he
addressed
has
as like a medical,
what's
you
that?
know, you got to be, it has to recognize that you're talking about medical information already.
It
But
must.
even yours got taken down. That one got taken down.
Yeah, the one with Vicky Mail got taken down, which I got it back up, but
Yeah.
still, yeah, I don't get it because Simon Dan, he has like hundreds of thousands of subscribers, and YouTube still dings him. It's very weird.
Yeah.
There was one
Off
that he
topic, but I haven't talked to you in a while. The natural origin of COVID it's in the can. It's done.
oh, it's done. It's dead. Yeah,
It's in. Yeah, I heard. What is it? Eddie Holmes, right?
Eddie Holmes. Yeah, yeah.
Yep, I heard him talk about that on one of his shows. He was like, "Well, we're going to look into the metatranscriptomics data." And I'm like, "Oh, well, there you go.
Mm-hmm.
There you go." And what did you find mostly? Not human DNA.
Yep. Yep. Yeah, they find samples in the Huanan market that are consistent with animals having carried the virus, and that is, um, I mean, there you go. You have that's literally the last like link that you
Yep.
needed to like without a shadow of a doubt prove that SARS-CoV-2 was in that market, animals were carrying it, and it was being spread to humans.
Right.
Because you can go further back in time, you can look for more infections that were like from earlier months. You just don't find them. So, and so the cases that they do find in like November, December, clustering around the market,
Yep.
it's why else? Why would a virus leak from a lab in a city of
Twice.
what 10 million people, 11 million people? Yeah, leak from
Hmm.
a lab twice, two
Go
different
30
lineages,
kilometers.
and then show up in all places in a market where you would expect a natural spillover to happen. Like
Oh yeah.
there's just no there's no way that it makes sense. Actually,
But while we're
I
still,
think I
but
told
while we're still talking about that, there are still markets. I guarantee you.
yeah, yeah. I mean those
That's the scary
markets.
thing.
There are still markets in China that are selling animals that could be carrying new viruses. I mean, it's we're gonna have another pandemic eventually. Might
Oh
not
yeah.
be a coronavirus, but we're gonna have another one eventually. Um, but yeah, I think I told someone a while back, like the only scenario now that makes sense. It still doesn't really make sense, but the only scenario that I think is even possible would be if they on purpose made the virus and on purpose planted it in the lab in the market. But that's just ridiculous. Like that's just
That's
way into conspiracy territory. Like there's
no.
absolutely no evidence for that. Like so it's just it's an idea that's 6 feet under. It's just
Oh
dead.
yeah. It's gone. No, not yeah, not anywhere in the scientific literature.
Yeah.
Never be. It will never come anywhere near this. It's done. The story is done. That's that's probably what it is. It's not going to go any further. There's no because all of the evidence is long gone at this point.
Yep. And I think like the virus deniers, the lab leak people, you know, they've been saying the same stuff for a long time now. And I think in a few years, it's still going to be the same stuff. They're still just going to
Hmm.
be saying, like, oh, but the emails and oh, but the lab is right there, and they're just not going to have
Yeah.
anything.
I would say, I would say like that insertion is a very unique insertion, but it's not in a region that's constrained, so you would expect to see an insertion there. The way it was changed was interesting,
Hmm.
right? But things change in interesting ways. That's
Right.
what it does. It forms, it's like saying, well, somebody shaped that water to look like the vase that it was put in. That's amazing. It's like, no, the water formed to fit the vase.
That's true. Yeah. Yeah, and also, I think other people have made this point before, but sarbecoviruses are not super well sampled. Like, we don't
Right.
know, we don't really know how unique a furin cleavage site like that is. We don't know how often it happens in nature, and we just don't sample it
And
because
it's the
the
what RB2 site, the one that
RB2.
matches like almost identically to pangolin.
Oh, yeah, yeah. Yeah, I mean, it's still fuzzy, like where exactly SARS-CoV-2, like what path it took
Hmm.
to get to humans. Um, but I mean, the sequence suggests pangolin, maybe, but we don't really know.
Right. And they
Pangolin was
didn't even
maybe
know
part
what
of it.
they didn't know what that sequence even looked like before
Right.
the pandemic. So yeah.
All right, man.
Okay. Yep.
It's
Time to
it's
go. Time to
almost
go.
it's almost 10:40. Should probably start winding down.
Yeah.
Does
Big
David Gorse?
congratulations on all the excitement going on in your personal life, home life.
Oh, thanks. Yeah, I hope things are going good with you. Should we
Oh
should
yeah.
have we should have a call where we just hang out sometime.
Yeah, that would be fun.
Yeah.
Just talk about the actual science that's going on. Yeah, lots of really great stuff going on in lab. A lot of really good things. So can't complain here.
Yeah.
That's for sure.
Yeah. I can talk about interesting things going on at work too,
Oh yeah,
but
cool.
yeah. All
Another
right,
time.
man.
Great
Um,
job today.
Thanks. It was fun. It was exactly how I expected it to go. Had no surprises, and hopefully, like I said, this helps someone who might be on the fence.
Yeah,
But
yeah.
all
That's the
right,
aim. See ya.
all right, everyone. Have a good night.
All right.
Thanks for watching.